Thank you for the very nice introduction. And thank you, Valerie, and the organizing committee for inviting me here to speak today. Also, thank you to the Chen Institute. As Yan mentioned, they've been tremendously supportive of our neuroscience center at Caltech, and particularly for me, the Brain Machine Interface Center. So some of what I'll be talking about today is funded under the Chen Institute. So I'll be talking about unlocking movement, helping paralyzed patients with brain-machine interfaces. And the outline of the talk, first I'll explain or give a little background on a brain-machine interface, in particular how it can read out brain activity to use the intent of a subject to control his or her environment. Then I'll discuss the idea of a cognitive neuroprosthetic. This is largely based on decoding from association cortex like the posterior parietal cortex. A couple of examples that I'll give are social observation and language. Next, as in the introduction, I'll be talking about writing in signals, in particular somatosensory signals, and this is for more dexterous hand movements. And then finally, I'll talk about some new technologies that we're working on. One is a less invasive functional ultrasound, BMI, and the second is an application for driving for tetraplegic participants. So this is the basic layout of a BMI. It starts with a recording or readout of neural activity from the brain. That goes to a decoder that decodes the meaning of the different patterns of activity. We can use those to control external devices like robotics or computer screens. And we can also write in signals, and this is by electrical microstimulation in the cortex. So the causes of paralysis are many. The one we concentrate on is spinal cord injuries, but of course these are approaches are applicable as well to stroke, MS, ALS, and Duchenne's muscular dystrophy. And the participants in our study have tetraplegia, so they have high spinal cord injuries. About half the subjects with a severed spinal cord, it occurs at high levels, at cervical levels 3, 4, or 5, 6, and that's our participants. And they all 4 limbs are paralyzed, and also all the feeling below the level of the lesion for touch and proprioception is lost. Now, usually these subjects have their lesions early in their lifetime. It's kind of when you're young and you do kind of reckless things or engage in athletics or so forth. And they survive for a long time. And as a result, there's a great deal of expense and loss of independence throughout the lifetime. It requires a caregiver. So if we could use these brain-machine interfaces to provide independence for tetraplegic subjects, it would both be great for their independence as well as a cost efficiency. Now, the subjects are the real heroes of these experiments. These are the subjects that have participated in our studies, and they do this not to help themselves, but rather for help for people down the road in the future. However, it does, although we don't mention it during the consent, I think it's turned out to be very valuable for our subjects as well. Paralyzed people often don't get out. They don't like the attention of being in a wheelchair. They become depressed. In our studies, they work for 5 to 8 years, 2 or 3 times a week for 3 or 4 hours. So they have a job. We pay them for their time. They have a goal and a mission. They get out to meet the researchers. We get to know their families. They participate in a lot of outreach functions if they're interested in it. So they've participated in programs at the Society for Neuroscience or outreach programs to other people with paralysis in the Los Angeles area. So overall, and I guess the most important thing, an important thing, which is the reason why many people go into clinical trials, is they have surgeons and doctors more or less on speed dial. And so they receive, as a part of the clinical study, great medical care. So this shows the basic problem with the paralysis with tetraplegia. Normally, you see an object, say, that you want to reach out and grab. That information is routed through the retina and into visual cortex, goes through posterior parietal cortex and prefrontal areas, and then from the motor cortex down to the spinal cord. And when you have a lesion in the spinal cord, it's broken this loop. But the subjects can still think about moving. And so the idea is to record that movement thought and use it to assist the subjects. And this is the type of electrode that we use. It's very common. Other BMI centers use it. Probably most of you are aware of it. It's a so-called Utah ray, and it has 96 active electrodes on it. And I'd like to emphasize in particular how big it is. It's only 4 by 4 millimeters. So it's a very tiny device that's getting implanted in cortex. This is an implant in this SMG, the supramarginal gyrus. You can see it's extremely small here. What we see when we're recording is like on the screen. So you can see the 96 channels, and we're just highlighting one of the channels. And we use as our features usually threshold crossings of spike action potentials. And this display shows the 96 channels in one particular implant. These are simulated neurons, but this is what we look for in our decoding. So the first neuron is tuned to movement left, meaning on here several trials, it has increased activity for moving left, not so much for moving right. And then, of course, you have the mirror neuron that's active for moving right, and other neurons that are active for other directions of motion. So the sort of magic is the decoding. And there are many really interesting decoding algorithms. Many of my AI and computational neuroscience colleagues are always working with us, coming up with things. But often the most simple is the best. And so in this case, this is just an example of a linear decoder. It's nice because we have very little data, small amounts of data, and we don't want to overfit. And so that works pretty well. So, and also it's the simplest to explain. So you have the data collection that you saw. Then we extract features into bins. So this is the different neurons and their spiking activity. And we add up the activity, in this case, usually 50 millisecond bins. During the training period, we fit a model to the neural activity. And then during the what's called closed loop period of the experiment or the session, we then use that trained decoder to interpret the intent of the subject. So these are the areas of the brain that we'll be concentrating on. The motor cortex is where most people go, and that makes sense because that's the area that sends out the command signals to the body. We do record there. We stimulate in somatosensory cortex, so that way we can return somatosensation to the subject. And we also highlight the posterior parietal cortex. So we're looking at that for what I refer to as a cognitive prosthetic, which can decode higher aspects of motor planning. So the posterior parietal cortex is a very interesting area. It's an association area, and so it connects many cortical areas. And it's also important for sensory motor integration. So if you look at the literature, both animal recordings and lesions in the clinic, you find that it's involved in many types of cognition. So among them is action planning. It integrates all the senses. So you see vision, you see touch, you see audition. It's important for aspects of language. It's also known to be important for navigation. People with lesions there have difficulty navigating within their environment. It's important for eye movements, reaching, and grasping movements. So it already shows to be a very rich area to decode a number of cognitive signals. And so this is just an example of one of our participants, and he's controlling a cursor here out of posterior parietal cortex. So what you see there is when the target turns red, he moves the little computer cursor to the red target, and it's a so-called center out task. It's usually kind of a basic cornerstone task that we use. And you can see that he can do this really, really well. In fact, as good as I can do it. I've tried it, but with my hand and a mouse. Also, when our subjects begin a task or begin a trial, we ask them, what's something you'd like to achieve? And so our first subject, Eric Sorto, he wanted to be able to drink a beer without his mother helping him. And so this is the first time he was able to do that successfully.
You finished that thing off? That was good. And that's how science is made. All right. Yay!
There you go.
So he was super happy about that as were we.
Did it.
Yeah. So you can see it's a lot of fun doing this kind of research. Uh, the so again, like I said, there are a lot of functions in posterior parietal cortex. What you saw was us decoding trajectories. We could decode the goals of a subject. Uh, but as we looked at it further, we could decode individual fingers, and on both hands, we could decode intended movements of the entire body, both sides. That was pretty amazing. We could decode different grasp. We could decode the observation of other people and the interpretation. We could decode aspects of language and also navigation tasks. We could decode navigation abilities. So the question becomes, and this was quite surprising to me and to all of us, was how is it that with such a tiny array, you could just plug it into one spot? Usually we put in more than one array, but not an awful lot of arrays. And you get maybe 100, 200 cells from a single array that you put in. How is it possible with so few neurons to be able to decode so many things? So that is kind of a good problem for us, but an interesting one. And so this is an example of observation. And what you'll see is that Nancy Smith, there'll be a cell that's active when her chin is touched, but not her cheek. And Deborah will be doing that touching. Carrie here will be touching Tyson's chin and cheek. And what you'll hear is that there's activity related to touching her chin and also activity related just to touching Tyson's chin as well, but again, not the cheek for either of them. So it's as if she not only feels the touch, but she can infer what the other person is feeling. So you can hear the neuron clacking away. And this is activity for touching the chin, not so much for the cheek. Or chin again. Tyson's asking Nancy to close her eyes, and you can still hear the activity touching the cheek. So that indicates it's not visual, it's actually touch that she's feeling. So what we thought we were seeing was an example of a mirror neuron for sensation. And how many of you have heard of mirror neurons? Everybody, almost, okay. And so this idea was first discovered by Giacomo Rizzolatti from Parma, and he and his group noticed that cells that were active during a monkey grasping an object, but also, so that and also if the experimenter grasps something. And so it shows specificity in that it would say be a grasp, and they had an example where they reached towards something but not grasp it. It also has congruency, which means that it's for both observation and for actual grasping. So those are sort of the two primary aspects of mirror neurons. And it has since been speculated that this is important for a large number of things like theory of mind. You can understand what someone else is doing by interpreting it through your own motor system. It's also been extended to language and other higher cognitive functions. So we thought that that might be what we're seeing, but we decided to look at it in more detail. And so we have, in our case, we have the two persons, the actual and observed persons. We use two body parts, the cheek and the shoulder, and four different touch types. Here, pinch, press, rub, and tap. And so that's 16 different variables we're looking at. And here's one neuron. You can see it looks like a mirror neuron in that it has activity for actual cheek and observed cheek, but not for actual or observed shoulder. But if we look at the different types of touch, we find that one of them is only active for pinch, the other is active for all four types of touch. So it doesn't have that specificity or congruency that we're looking for. And this is kind of the simplest example. Here's some other neurons, and you can see that this cell here is only active and has tuning for the different touch types only for actual cheek, not for any other body part or observation. This one only for pinch, but for observed and for shoulder. So it's active across these different contexts. And here's one that's only active for observed cheek, and it shows tuning for the different types of pinch. So these don't look like mirror neurons per se. And I think even in the original descriptions, there was only about 12% of neurons that fit the mirrored neuron description. And as you start to look at more and more variables, fewer and fewer cells look like mirror cells. So it's not saying that there isn't this mirroring occurring in this area, but rather that it's occurring at a population level. So you can read out what the ideal mirror neuron is doing at the population level, but not necessarily at the single neuron level. So why is this important? Well, it, I think, begins to allow us to touch on why so many things can be decoded from such a small population of neurons. And one concept is that it's coding what we call, and others, mixed selectivity. And so if you plot, say, an activity of three neurons, so that the axes each represent the activity along one cell, and you might find that for actual rub of the shoulder, that these neurons will show different levels of activity that are specific for that particular stimulus. But for another, here imagine rub on the cheek, the same three cells will be active. So they mix the information, but the activity is different. So you can, within this neural space, you can separate those two activities. And here, just with three cells. And a second concept is compositionality. So this is very common in language, and it's the idea that you have basic building blocks within the population code. So if you had, say, a concept for the meaning of bald and the concept of meaning of man, then you could combine these two building blocks into meaning of a bald man. So looking again at the population code, if we do a demixed PCA, so basically we're looking at the variance that occurs with our three variables, that being body part, touch type, and person, we're seeing where we would have a high degree of variance where we could then decode that particular activity. And they seem to fall out into these building blocks. So you have a high degree of variance explained by body part, a high degree by touch type, and a high degree by the person. And another way we can look at this is cross-decoding. So that's looking at the amount of shared or information within the population code. And the way we do that is we'll say within the space of the touch type and the shoulder cheek, we can look at actual and we can test. So in other words, this would be our test data, and then we can look at how well it could, so we're looking at this from that subset of populations activities. We look at what matrix of weights we would get to decode the body part and the type of touch. And then we test it, we train it here, and then we test it on the observed. And what we can see then, for instance, with this particular slicing of the data, that we can generalize to body part and touch type. And if we look at the other splits of the data, we also can do that. So again, this fits with the idea that there are these, within the subpopulation, there are blocks that code for touch type, for body part, and for person. So taking these two ideas together, partial mix selectivity and compositionality, we can imagine how you might be able to, in this small population of neurons, pack all that information in there. Okay, so that doesn't mean, though, that there's no organization in posterior parietal cortex. Actually, there is. I've been talking about activity record from the superior parietal lobule. Recently, we've been looking at grasp in the inferior parietal lobule, and somewhat by serendipity, we noticed that we could also decode speech from that region. So if we could decode speech, and you'll hear tomorrow about some elegant experiments decoding speech from frontal areas, this would be enormously useful for subjects that have dysarthria. That is, they, like in ALS, they are so, locked-in subjects, they're so paralyzed that they can't even speak. So if they could think about speaking, imagine speaking, this would greatly help for them to facilitate communication once they're in that state. And so there's been, speech is a very hot area now in BMI, BCI research, and groups have shown that they can decode attempted speech and also mime speech. So this is where the articulators move, but no sound is made. It's, but it hasn't been shown yet, well, until I'll show you some data for this, internal speech. So this is the inner dialogue that you have with yourself. And so in that case, you're not actually speaking, but you're imagining speech. So how many people have inner dialogue? Okay, good. So it usually breaks down like that, 80-20. So it's a very personal thing, I guess. And one interesting thing about doing this kind of research is that there may be differences, individual differences between people. So we also don't think that we're decoding a person's internal thoughts. So to do that, they would have to follow along in the task and do the requested internal speech. So the task we use, we begin with an ITI, intertrial interval. There's a cue then which can either be written or a sound. So it could be Python written or speaker producing the sound Python. Then there would be a delay, a cue to have internal speech, another delay, and then a then a cue to actually speak the word. And if we, these are the words we used. There are 8 of them, 6 words and 2 pseudo words. And these are the data collected. And you can see that for the cue, we can classify with, again, this is chance here, a pretty good degree of which word is up there. There's still some classification within the delay period as the activity goes down. Then with the inner dialogue, we can decode the words they're saying to themselves. And then with online decoding, which I'll show, oh, I'm sorry, speech, we're up here about 74% actual speech. And then we also have an online decoding task, and this is where I'll show you in a minute the subject gets a cue, and they then think about the words, and then we read out what we think they're thinking. And with the online performance, if we train with 16 to 20 examples, we get pretty high performance, around 79%. Okay, so this is the online task, and there's the word, the subject reads it, there's a cue to say the word internally, then we decode that internal speech, and then we provide them feedback. So if it was swimming, they get swimming if that's what we think they're thinking, and it comes back with green characters, and if it's a different word, we show the word and it's in red. Okay, so this is the subject sees swimming here and then gets the cue to say it internally. So that's the internal speech. And then we decode swimming. So we got it on that one. And here's a set of 3 more, 2 successful and 1 miss. Okay, so now in terms of writing in signals we do that for manipulation. So, and to show why it's important to have these somatosensory signals, this is from Johansson. So it shows the subject striking a match, and then her fingertips are anesthetized, and now she tries to strike a match again. And so this demonstrates how important it is to have sensation when doing fine manipulation. So with our paralyzed subjects, they can visually guide and get visual feedback to grab something. But once they've grabbed it, in principle, they won't be able to manipulate it because they have no sensory feedback. So to get around that, we've sensorized robotic hands, and we can, the idea is then when the robotic hand touches something, that stimulation is delivered in the cortex to provide that feedback. And you all are probably familiar with the homunculus on the somatosensory cortex. Here is 2 implants that we made in the hand representation of somatosensory cortex. The dark colors just represent electrodes that produce the touch sensation. We can see that array 1 here is best for middle and ring finger, and this follows the somatotopy 2 here for middle and index finger. And just as an aside, this is pretty amazing because these subjects have been paralyzed for many years, and yet on the first day when we stimulate, they feel it, and they feel it in the right place. So at least in adults, these representations are highly crystallized and available to us. And that's actually a good thing because we thought there might have been a high degree of plasticity. For instance, the face representation might have moved into the limb representation, in which case they'd be feeling the stimulation on the face and not on the hand. So this is an example of a subject who's he's now in virtual reality. He's touching this little blue ball here, and you can see the activity occurring here, and when he touches it, you'll see some stimulus artifact on the screen as well. So there's the artifact coming in. So now we're in the process of combining these two to make hopefully more dexterous manipulation. Now, a new technology that we're exploring is functional ultrasound. It's a less invasive form of a BMI. So there are a variety of technologies, EEG, COG, so forth. This is the new one, and it's a relatively new imaging technology. It was developed here in Paris by Mikhail Tanter, and the idea is to have ultrafast ultrasound coming down in sheets instead of as a focused beam, and then the reflections coming back and being analyzed. So we've been collaborating with his lab as well as Mikhail Shapiro's lab, and advantages of using this technology are one, that it has a very wide field of view. So you can see that we can get, in fact, centimeters. Am I getting close?
Yeah, we're close to the ending time, so
Okay.
you want to save some time for questions.
Oh, okay. Yeah, so I'll go quickly. So the another nice feature is that instead of implanting the ray directly into the brain, where you have to make a dura flap and punch the electrodes into the brain, you could actually, the skull impedes ultrasound, but you can put a little window in there. And so that's a much less invasive type of surgery. And so this is an example of a monkey where we're decoding 8 directions of eye movement. He doesn't actually move his eye, he just has to think about it. And these are voxels that just show how well they're tuned. So you notice that the percent change in this case, power Doppler, is up to about 40% compared to just a few percent for MRI. And also it's highly stable, so that this is just an example of decoding training with, I think, 80 or so trials the first day. The next day, you just need 13 trials of training. And it's because you're looking at an image, so it's very stable. And so that's a nice feature. And we've now transitioned this to humans, where people with traumatic brain injury have to have pressure relieved by removing part of the skull. And so you can see the surgery here. The surgeon, Charles Liu, replaced the skull with a plastic that's transparent to ultrasound. And so here the subject is resting in this case and then playing guitar here. And you can see the activations, looking at the pixels. It's pretty obvious you could decode those movements of the fingers. We've also got a project on driving, This is with Ford Motor Company and with BlackRock. These are just some simulations of the subject. So one thing they're worried about a lot, is being able to brake quickly. Our subject can brake at the high end of what normals can. We think it's because it's coming directly from his brain. And this is just a little video that they made. I guess at some point it'll be a commercial or something.
Just fine tune it.
Are you trying to stay in a lane, or?
Yeah, I'm trying to stabilize it. We're doing pretty good. Getting behind the wheel again, actually sitting in the front seat, in the driver's seat, that's gonna be pretty awesome because it's been almost 5 years now. And 5 years of sitting in the back seat, being at the mercy of my teenage son driving, it'd be great to finally have some control for myself.
So we're working with Richard Murray's lab, who works on automated driving. We'll use prediction and error signals that are in posterior parietal cortex, as well as navigation representation there to expand on this. So in summary, things are coming along well. As I mentioned, the participants are the real heroes. We thank them. The funding, the TNC Chen Brain Machine Interface Center, and various grants. We collaborate with the laboratories at Caltech, medical schools around Los Angeles, as well as rehab centers, and the Tantra Lab in Paris, and a company in Paris, Iconius. And those are the current members of the lab. On the right, you saw the pictures of and names of others that have worked on these projects in the course of the talk. Okay, thanks.